So yes, very good evening to all the NEET aspirants 2026 and 2027 as well. Here we are to discuss the 100 concepts for NEET mega sessions, where we have planned most 100 concepts from all the chapters of botany and few of from zoology as well, like genetics and cell biology and all. So let us discuss it in more efficient way. As such, today we are going to discuss about mitosis. So before that, I would like to introduce unique programs in our NEET world with the most minimal and affordable price, wherein under 2 steps to medicine, which is aiming for 11th standard students, current 11th standard students who are aspiring to write NEET 2027 with the very minimal free fee, that is 3333 per month, okay, including the complete material for NEET, okay. And 1 step to medicine, which is for 12th standard students with fee of 3333 to the students who are aspiring for NEET 2026, and the long term students, they have a special program, that is power step to the medicine under NEET world with the same fee 3333. So it is same and reasonable for all the students per month, beta. Make use of it, okay. And the new batch here in offline is going to start. This is going to start from July 1st, 2026. What are the unique features of this course? Like live and online sessions both are available, where online classes you can access in MedWorld app. Just you can install this app in the Play Store and you can log into the course. You will get into the complete classes. So classes will be in the hybrid mode and full syllabus coverage with NCERT and extra concepts, and with complete objective practice as well. Well crafted material will be given, one-on-one mentorship, and here in our offline branch Narayanguda, complete digital AC classrooms will be there. Hostel facility is available both for boys and girls. So whoever are interested, just call us on the given number, and the fee is, once again remember, the fee is very minimal, that is 3333. This offer is for very limited, okay, and the seats are also limited, so make use of it. Yes, now getting back to the concept, that is mitosis from cell division. Most of you might be thinking, yes, mitosis is very easy, but then there are many important points you have to remember with respect to mitosis, and each and every sequential change that is happening in the steps of karyokinesis, be it prophase, metaphase, anaphase and telophase, everything you have to remember. And cytokinesis also, plant cell cytokinesis, animal cell cytokinesis, everything we will be discussing now by applying some. Lastly, we are going to practice some numerical questions also. Firstly, mitosis or M phase is having 2 subsequential phases. One is karyokinesis, we will call it as karyokinesis, and the phase after karyokinesis is called as cytokinesis. Mitosis is also called as equational division or somatic cell division. It is called as equational division because the number of chromosomes are same in the parent cell and the daughter cell. As the number of chromosomes are same in the parent cell and daughter cell, we call it as equational division. Somatic cell division we call it as because it occurs in the somatic cells and even in some gonad cells, which are in germ cells also, like some gonad cells. But as it is purely sticked or occurs in the somatic cells, this is also called as somatic cell division. So as such, we have karyokinesis under metaphase, karyokinesis and cytokinesis. Karyokinesis we have prophase, metaphase, anaphase and telophase. We will call mitosis as the most dramatic phase, and there are no clear-cut lines that we can draw for prophase, metaphase, anaphase and telophase. There we cannot draw any clear-cut lines for pro, meta, anaphase and telophase. Everything happens in a sequential way, in a progressive way. But then for our clear understanding, we have sequenced it under 4 steps for karyokinesis. Understood, ma? Let us learn each step in detail. First is what is prophase, right? Under karyokinesis. What is meant by karyokinesis? Karyokinesis is nothing but nucleus. Karyo means what? Nucleus. Kinesis is movement. Nucleus movement. Karyokinesis. So yes, in karyokinesis, the very first stage, that is prophase. First is what? Prophase. In prophase also we can nicely arrange that into early prophase and late prophase. In early prophase what happens is, all the chromatin thread or network like structure is attached to the nuclear membrane. This is called as what? Spireme stage. Okay, this is spireme stage. And slowly the chromosomes with chromatid structures with 2 sister chromatids are formed. Sister chromatids. And parallelly this happens inside the nucleus. Inside cytoplasm what happens is, 2 centrioles will radiate towards the opposite poles. We know centrioles which are perpendicular to each other, right? So 2 centrioles along with the spindle fibers, they will radiate to the opposite poles. So here you can see spindle fibers plus asters. Asters move to opposite poles. Okay, spindle fibers and asters, where asters move to the opposite poles. Spindle fibers and asters form spindle apparatus. Clear, ma? And at the end of the prophase what happens is, lastly at the late prophase, at the end of prophase what happens is, nuclear membrane, nucleolus, endoplasmic reticulum, Golgi bodies will be degenerated. Clear? So what are the sequential changes that happen in early prophase and late prophase? In early prophase, the chromatin material will be attached to the nuclear membrane. You call this as what? Spireme stage. Then untangling of this chromatin material leads to the formation of chromosome, where 2 sister chromatids will be formed. And then parallelly in the cytoplasm, you can see spindle fibers and asters radiating out to the opposite poles. Thereby spindle apparatus will be formed. Clear? And then at the end of the prophase, in late prophase, complete nuclear membrane, nucleolus, endoplasmic reticulum and Golgi bodies will be degenerated, and you can observe dense cytoplasm. These are the activities with respect to prophase. Clear, ma? And the second most important phase, that is metaphase. Okay? Second most important phase is what? Metaphase. In metaphase, chromosomes will be clearly visible under the visible microscope. Okay, under the light microscope. Chromosomes are clearly visible under the light microscope during which phase? If that is the question, your answer should be what, ma? Metaphase. And in metaphase you can see, chromosomes are thickest and shortest. Chromosomes are thickest and shortest. And the structure or the morphology of the chromosome is clear. Structure or morphology of chromosomes is clear, visible. Clear, ma'am. So, the chromosomes are thickest and shortest. The structure or morphology of the chromosomes is clearly visible. And now, in metaphase, all the chromosomes which are present at different spaces in the cytoplasm, they will come to the center point, where if these are the asters, in metaphase what happens is all the chromosomes will come at the center, forming an equatorial plate, and the spindle fibers are attached to the kinetochores, which are disc shaped structures around the centromeres. To this kinetochores, spindle fibers are attached and asters are at the opposite poles. Okay, asters are at the opposite poles like this. Okay, asters will radiate out the spindle fibers. They will be connecting to what part of the chromosome? Kinetochores. These are the disc shaped structures on the either sides of the centromere, whereby the centromeres or the chromosomes are arranged in the equatorial plate. This is called as, this phenomenon of chromosomes arranged on the equatorial plate is called as congregation. And then in anaphase 1, what happens is this centromere will be split, and then this half of the chromosome will be moving towards the lower pole and this side of the chromosome moves to the upper pole. Let us see what happens in the anaphase. During anaphase of the mitosis, third is what? Anaphase. So, with regards to metaphase, what you have to remember, ma? Thickest and shortest chromosome. You can clearly see the structure, morphology of the chromosome under the microscope, and equatorial plate. The phenomena of the chromosomes landing onto the equatorial plate is called as congregation. Just a second, here is a So, this is called as congregation. Next, anaphase, where in anaphase you see centromere splitting is the major phenomena that is observed in the anaphase. What is observed in the anaphase? Centromere splitting. Centromere splitting is observed in the anaphase. Because of centromere splitting, you can see different shapes of the chromosome. Centromere splitting will give different shapes of the chromosomes. Shapes of the chromosomes. The shapes can be V, L, J and I, wherein V, two arms of the chromosomes are equal like this. That is called as metacentric chromosome. Okay. Two arms of the chromosomes are equal in length, that is metacentric chromosome, and centromere is at the center. Okay, centromere is at the center. You have two equal arms of the chromosomes. This is called as metacentric chromosome. This is called as metacentric chromosome. Metacentric chromosome. Clear? So, here as two arms of the chromatids are equal, you call it as isobrachial chromosome. Okay, this is isobrachial chromosome. Clear, ma? Next, L shape if you have to observe, centromere is close to one arm of the chromatid and it is far from the other arm of the chromatid. As a result, you can see one short arm and one long arm. Short arm is called as P arm and long arm is called as Q arm. So, here you can observe, this is called as submetacentric chromosome. Take down beta, this is very important. This is submetacentric chromosome and you call it as heterobrachial chromosomes. These are what? Heterobrachial brachial chromosomes. Fine. Next, J-shaped, you can see one extremely short arm and one extremely long arm. Okay, one extremely short and one extremely long arm. So, this extremely short arm is what? P arm, and this is Q arm. Same case, this is the heterobrachial chromosome, but you call it as acrocentric chromosome. Acrocentric chromosome. Clear. Next, I-shaped, wherein one arm of the chromatid is very negligible, that is very small, whereas the other arm of the chromatid is long. This is called as telocentric chromosome. This is called as telocentric chromosome. Clear, ma? So, here you can identify after centromere splitting what happens. Different shapes of the chromosomes are observed, like V-shaped, J-shape or L-shape or I-shape, and all the chromosomes will be close to the poles. Chromosomes are close to the poles, close to the asters, you can say. So, here if you observe, the spindle fiber length will be reduced. Spindle fiber length is reduced. Spindle fibers length is reduced. Spindle fibers and chromatids are close to the asters. Remember the sentences clearly so that you should not get confused in the statement based questions, beta. Okay. So, here spindle fibers length will be decreasing and chromosomes and spindle fibers are close to asters. Clear, right? This is all about anaphase, where the major event that happens in the anaphase is what? Centromere splitting. Once the centromere is split, the two arms of the chromatids form different shapes. They will segregate towards the asters in different shapes. It can be V, L, J and I. V shape, where it has two equal arms of the chromatids, you call it as isobrachial or metacentric chromosomes. L shape, where short arm is called as P arm, long arm is called as Q arm. This is submetacentric, and as two arms are of unequal length, you call it as heterobrachial chromosome. J shape also it is same, but it forms extremely short and extremely long arm. This is acrocentric. In telocentric chromosome, which is I-shaped, one is negligible arm and one is long arm, that is telocentric. And here in the anaphase, chromosomes and spindle fibers are close to the asters. It is as simple as it is. This is all about anaphase. In telophase, the major event that occurs in the telophase. Telophase, what happens is, again the chromatids which are present, where let us suppose 3 chromatids over one end and 3 chromatids towards the other end of the asters at the end of the anaphase. So, here tangling of the chromosomes takes place. Chromosomes get tangled. Chromosomes get tangled to chromatids again. Fine. Chromosomes get tangled to chromatids again, and reappearance of nucleolus, reappearance of the nuclear membrane, endoplasmic reticulum, Golgi bodies occurs in the telophase. So, in telophase what happens, beta? Reappearance of nuclear membrane, nucleolus, and endoplasmic reticulum, Golgi bodies happens at the end of telophase. So in telophase, that is as simple as it is that you see during prophase, that is exactly opposite to the prophase, wherein in prophase chromosomes get untangled and they form chromosomes, okay, where the chromatin material gets untangled forming chromosomes, but here chromosomes get tangled back to the chromatids. At the end of prophase, you see the nuclear membrane, nucleolus, endoplasmic reticulum, Golgi bodies will be degenerated, but here in the telophase everything will be reappeared. So this is all about prophase, metaphase, anaphase and telophase. Once let us quickly recall that in the interphase, after interphase, during early prophase, what we have discussed beta? We have discussed about spireme stage, we have discussed about chromosome formation, sister chromatids, we have discussed about asters that are moving to the opposite poles and spindle fibers connecting to the kinetochores of the chromosomes. And at the late prophase, what happens? Nucleolus, nuclear membrane, Golgi bodies, endoplasmic reticulum will be degenerated and there will be dense cytoplasm. In metaphase, what happens? All the chromosomes come to the equatorial plate. This is called as congregation, and spindle fiber length from asters will be increased, and structure or morphology of the chromosome is clearly visible in the metaphase. In anaphase, the centromere splitting will happen and chromatids will move to the opposite poles by spindle fibers, and this forms out different shapes of the chromosomes V, L, I, J. We have discussed both about isobrachial and heterobrachial chromosomes. In telophase, you can see all the nuclear membrane, nucleolus, Golgi bodies, endoplasmic reticulum will be reappeared. Clear? And here you can see the chromosomes get tangled again. Fine? The chromosomes get tangled back to the chromatin. So this is all about the continuous phases from early prophase to telophase. Now the karyokinesis part has completed. Let us look into the cytokinesis of both plant cell and animal cell. If you look into the cytokinesis of plant cell beta, what happens is, in a plant cell, the very first layer that is formed is called as phragmoplast. So very first layer that is formed at the center, that is called as phragmoplast. First formed layer is phragmoplast. Phragmoplast is secreted from Golgi bodies. Okay, this phragmoplast which is formed at the center will move towards the periphery, from center to periphery. This movement, it forms at the end cell plate, and this movement it called as centrifugal method of formation or centrifugal movement of cell plate. Okay, first initially phragmoplast is formed, which moves from the center to the peripheral sides, to the either sides, forming a cell plate which is centrifugal, and this cell plate turns out to be what? Middle lamella. This turns out to be middle lamella, which is the first formed layer of the plant cell. This is first formed layer of the plant cell and plant cell wall. This is the first formed cell wall of the plant cell. Fine. And this middle lamella consists of calcium pectate and magnesium pectate. This consists of calcium pectate and magnesium pectate. So in plant cell, how cytokinesis happens beta? First, initially at the center, phragmoplast is formed. Phragmoplast is produced from Golgi bodies that forms the cell plate, which moves from the center to the peripheral ends. This movement is called as what? Centrifugal. As such, the initially formed cell wall is what? Middle lamellae. Middle lamellae is the first formed layer of the plant cell wall. This is made up of calcium pectate and magnesium pectate. Remember that. But exactly opposite happens in the animal cell. In animal cell, wherein cell furrow is formed from the peripheral ends to the center. Okay, cell furrow is formed from the peripheral ends to the center. So this movement is called as centripetal because it is from the peripheral ends to the center. As such, two equal cells are formed. Two nucleus moves to the opposite ends. Cell furrow will divide these two cells. Clear? So this is about cytokinesis. Clear ma? Cytokinesis in both plant cell and animal cell. Fine? Now, significance of mitosis. What is the significance of mitosis? Primarily growth of organisms, where mitosis increases the number of cells and enables the growth in multicellular organisms. We can define growth in multicellular organisms as increase in cell number, right? So mitosis will increase in cell number. Second is tissue repair and regeneration. Okay, to repair the tissue, lost tissue and regenerate, mitosis is the only way. And for asexual reproduction or vegetative modes of reproduction in plant cell, through mitosis only it will happen. And here one more point I have to say you, that mitosis happens only in the diploid cells of animals. In animal cells, mitosis occurs in the diploid cell. Whereas in plant cells, mitosis occurs both in the haploid cells and diploid cells, which means in the haploid, gametophytic cells and diploid, sporophytic cells. Clear ma? So one best example is during the development of female gametophyte, that is embryo sac. Okay, that embryo sac is developed from what? After 3 sequential mitotic divisions of megaspore. Megaspore undergoing 3 sequential mitotic divisions, that forms a female gametophyte, that is embryo sac. So haploid megaspore giving rise to haploid embryo sac. Haploid megaspore to haploid embryo sac. Clear? So this is the best example for mitosis in the haploid cells. Clear? Next, genetic stability. Mitosis will maintain the genetic stability all across the generations, from parent cell to daughter cell. Next, prevents the cell outgrowth. Okay, where mitosis is regulated in a balanced cell number, in its misregulation can lead to what? Cancer or tumor. Fine. Next, development from zygote. From zygote to embryo to fetus, that development happens through mitosis only, and it is mitosis is used in wound healing, during regeneration of the lost tissues or cell replacement, and this will control the chromosome number. These are the uses of mitosis. Now let us exercise 2 major numericals with regards to mitosis, wherein first, for the cell number of mitotic divisions. How can you calculate the number of mitotic divisions? By the formula n-1. And for bacterial generations, it is 2 to the power of n. Let us exercise both of them. First, using the formula n-1 ma. A tissue sample is found to have originated from a single cell, and this contains 1000 identical cells. So total number of cells are how many beta? 1000. Assuming the mitotic division and simultaneous splitting, how many mitotic divisions occur? From total cells, what is the number of mitotic divisions formula? n-1, where n is the number of daughter cells. So here number of the daughter cells are 1000 minus 1. It is what ma? 999. So this will be the answer. It is as simple as it is. Okay. Second, for number of generations, just focus here. A single bacterium every 20 minutes by binary fission. So this single bacterium will divide for every 20 minutes. One bacterium will divide for every 20 minutes. How many generations will occur in 3 hours? In 3 hours, how many generations will occur? 3 hours means how many minutes beta? 180 minutes. How many generations he is asking, not how many divisions. He is asking you how many generations and how many bacteria will be produced. First let us look into how many generations. That is just by crossing this 180 by 20 will give you 9 divisions. 9 divisions means what, ma? 2, 3, 4, 5, 6, 7, 8, 9. So this stands out to be one generation, second generation. As such, for 9 divisions you can get how many generations? 8 generations. How many generations, beta? 8 generations. So in this 8 generations, we have to substitute in the formula 2 to the power of n, where n is the number of generations, where 2 to the power of 8 will obviously give you 256. So at the end, how many bacteria are produced? 256 bacteria. Clear? Simple application. First you see one bacteria, time of division is 20 minutes. And he said how many generations and bacteria will be produced at the end of 3 hours. 3 hours means how many minutes, ma? 180 minutes. Just cross it, you will get number of generations. After getting number of generations, substitute it in the formula of 2 to the power of n, then you will get number of bacteria. And one more question, look into here, one more question wherein, for example, a test tube is there, ma. 1/4 of the test tube is occupied by bacteria, wherein each bacteria division time is 10 minutes. How much time it will take to fill the tube? To fill tube, how much time it will take? Okay, this will be the question. It is very simple. So I said 1/4 of the cup is filled, and from this bacteria, one bacteria will divide in 10 minutes. Anyone from online can answer, ma? See, I will say, you look here, wherein each bacteria division time is 10 minutes. So all the bacteria in this 1/4 space, when it divides for extra 1 minute, it fills half of the cup. Yes or no? It fills half of the cup, and plus 1 minute of division will fill the tube. It is as simple as it is. So total in 12 minutes, it will fill the test tube. It is very simple, right? Whenever half of the cup or full of the cup or 1/4 of the cup is given, that it has this much of bacteria and each bacteria division time is 10 minutes. So if such questions are given, just if all the bacteria in this 1/4 space divides for 1 minute, it fills half of the cup. If it divides for 1 more minute, it fills the full cup. It is logical, okay? So like this you have to calculate, ma. These are the 3 possible numerical questions that you can get from mitosis. So everything is clear with mitosis, right, ma? We have discussed 100% of the concept today with respect to mitosis. We have discussed all the 4 stages of karyokinesis, events that happen in all the 4 stages. We have looked into the significance of the mitosis, and at last we have solved the numerical based questions and application based questions from mitosis. Now I would like to end this session by saying you the same thing, giving you the best possible advantages from NEET World, your step to medicine, where we are launching 3 major programs under 2 steps to medicine for currently 11th studying students, and 1 step to medicine for NEET 2026 aspirants, power step to medicines for long term students, where the complete fee for any batch will be 3333. Make use of this golden opportunity, ma. And we are here to tell you that the second long term batch is going to start from July 1st, 2026 with all these facilities and complete study material provided in teaching modes onto your comfort, online or offline. You can just call us to the given number and the fee will be 3333. Saying this, I would like to end this session. Thank you.